Hi everyone, I am Dr. C.S. Kulkarni, MD, and I will teach medical biochemistry. Before starting the topic, please like, share, and subscribe the channel. In the last session, we dealt with theories and activities of enzymes. In today's session, we will deal with enzyme kinetics. Velocity or rate of enzyme reaction is assessed by the rate of change of substrate to product per unit time. In practice, initial velocity is determined. In much time is allowed to lapse, the velocity may tend to fall due to decrease in substrate concentration below a critical level. The velocity is proportional to the concentration of reacting molecules. A plus B gives C plus D. If concentration of A or B is doubled, the rate of reaction is also doubled. If the concentrations of A and B are doubled together, the velocity becomes fourfold. V is directly proportional to A into B. At equilibrium, forward reaction and backward reaction are equal, so that A plus B bidirectionally gives C plus D, where A stands for constant of forward reaction and K2 stands for the constant of backward reaction. Forward reaction R1 is equal to K1 into A into B, and backward reaction gives K2 into C into D. At equilibrium, R1 is equal to R2, or K1 into A into B gives K2 into C into D, or K1 upon K2 is equal to CD upon AB, which is nothing but equilibrium constant. The equilibrium constant of the reaction is the ratio of reaction rate constants of forward and backward reactions. At equilibrium, forward and backward reactions are equal. Equilibrium is the dynamic state. Even though no net change in concentrations of substrate and products occurs, molecules are always interconverted. The numerical value of the constant can be calculated by finding the concentrations of substrates and products. If equilibrium concentration is more than 1, the forward reaction is favored. In such instances, the reaction is spontaneous and exothermic. Concentration of enzyme does not affect the equilibrium constant. Concentration of enzyme certainly increases the rate of reaction, but not the equilibrium constant or the ultimate state. In other words, enzymes make it quicker to react in the equilibrium. Catalysts increases the rate of reaction but do not alter the equilibrium. Coming to factors influencing enzyme activity, these are 1. Enzyme concentration, then substrate concentration, product concentration, temperature, hydrogen ion concentration, presence of activators, presence of inhibitors, presence of repressors or derepressors, coherent modification. Coming to the first, enzyme concentration. The rate of reaction or velocity is directly proportional to the enzyme concentration when sufficient substrate is present. Velocity of reaction is increased proportionately with the concentration of enzyme provided substrate concentration is unlimited. In this graph we can see that enzyme activity is plotted against velocity, where substrate concentration can be shown as plotting straight line with a slope. Hence, this property is made use of in determining the level of particular enzyme in plasma, serum or tissues. known volume of serum is incubated with substrate for the first time for a fixed time, then reaction is stopped and product is conjugated, which is called endpoint method. Since the product form will be the will be proportional to the enzyme concentration, the latter could be acid. Second is effect of concentration, substrate concentration. In a series of test tubes, equal volume of enzyme solution is taken, but increased quantity of substrate is added and the reaction is assayed in each test tube. The velocity is expressed in micromoles of substrate converted per minute. If the velocity is plotted against the typical concentration, a typical curve is obtained. A substrate concentration is increased, the velocity is also correspondingly increased in the initial phases, but the curve later flattens. In this graph, we can see that substrate concentration is plotted against velocity and the effect of substrate concentration is seen. Factors influencing enzyme activity. At lower concentrations of substrate, that is point A in the curve, some enzyme molecules are remaining idle. At substrate, as substrate is increased, the increased enzyme molecules are working. At half maximum velocity, 50% are enzymes are attached with substrate, that is point B in the curve. As substrate increases, all enzyme molecules are saturated, that is point C in the curve. Further, increase in substrate cannot make any effect on the reaction velocity, that is point D in the curve. The maximum velocity obtained is called Vmax. It represents the maximum reaction rate attainable in presence of excess substrate, that is at substrate level saturation level. In this graph, we can see that substrate concentration is directly plotted against velocity, and the maximum velocity is at point D, and after D, the curve flattens. Enzyme molecules shown as half circles, and substrate molecules are red dots. Coming to the Michaelis constant. According to Michaelis theory, the formation of enzyme substrate complex is as reversible reaction, while the breakdown of the complex to enzyme plus product is irreversible. In this diagram, we can see that substrate concentration is plotted against velocity, where enzyme concentration on Km is studied. Km value is substrate concentration expressed in millimoles per liter at half maximal velocity, and it denotes that 50% of enzyme are bound with substrate molecules at the particular substrate concentration. Km is independent of enzyme concentration. If enzyme concentration is doubled, the Vmax will be doubled, but the Km will remain same, exactly same. In other words, irrespective of enzyme concentration, 50% are bound to the substrate at that particular substrate concentration. Km is the signature of the enzyme. Km value is thus a constant for an enzyme. It is the characteristic feature of a particular enzyme for a specific substrate. The affinity of an enzyme towards its substrate is inversely proportional to dissociation constant, that is Kd, for enzyme substrate complex. E plus S bidirectionally gives ES complex, where K1 is for the forward reaction and K2 is for backward reaction. Then the ES complex with the constant 3 K3 forms enzyme plus product. Where all these are present, Kd is equal to K2 upon K1 and Km is equal to K2 into K3 upon K1. Therefore, the decrease in the tendency for the dissociation complex, the increase in the affinity of the enzyme for the substrate is seen. Km allows the affinity of enzyme of for substrate. The decrease of the numerical value of Km, the affinity of the enzyme for the substrate is increased. To cite an example, Km of glucokinase is 10 millimole per liter, while that of hexokinase is 0.05 millimole per liter. Therefore, 50% of hexokinase are saturated even at the lower concentration of glucose. In other words, hexokinase has more affinity for glucose than glucokinase. Double reciprocal plot. Sometimes it is impractical to achieve high substrate concentration to reach the maximum velocity conditions. So half Vmax or Km may be difficult to determine. Then the experimental data at lower concentrations is plotted as reciprocals. The straight line thus obtained is extrapolated to reciprocal of Km. This is called Line-Weaver-Burk plot or a double reciprocal plot, which can be derived from the Michaelis-Menten equation. If we plot 1 upon V against 1 upon S, it will give a straight line graph. Intercept in x-axis is minus 1 by Km, from which the Km can be calculated. In this graph, we can see that 1 upon S is plotted against 1 upon V, where minus 1 by Km value is obtained at the negative side of the x-axis. Here ends our session. Kindly like, share and subscribe the channel. Thank you.